I want to thank everybody that are present even today, and I want to thank our last facilitator about this RNA, single-cell RNA data analysis. Very exciting. Last class, last period that we had our training, I discussed about visual screening, then molecular docking, and I told us that we can dock either peptide with protein, DNA or RNA, so it's possible. Even VINA can also dock a ligand with DNA. VINA can do that too. And I try to introduce us to virtual screening. That is, if you have a particular protein, let's say for the previous facilitator that just finished, you have a particular protein, maybe from a particular point of subset of your cell, but you now want to say, okay, if that is that protein that is really causing the problem, okay, you can do visual screening to see, okay, if that is the real target. You can do visual screening to find out those existing drugs that could bind even to that particular protein. That is, you don't have idea of what that protein could do. So you can do visual screening to find the ligand that can bind to that protein. After that, you have been able to find, okay, different set of ligand that can bind to it. Then from there now, okay, if you want to now design a new set of drug, then you can design it using the scaffold of all those ligand that you have discovered. And if not, then from there you can carry out your analysis. So you don't need what is it called now? Let's say, for example, okay, after you have done the visual screening, you're able to find some approved drug. So the next thing you can do, okay, you want to take it further, okay, let me take it to molecular dynamics simulation, okay, to see if truly all this ligand or all this drug could binds to this my protein. So after you have done molecular dynamics simulation and everything seems okay, then you can go back to the wet lab. And also, last class, I discussed about how to use VINA, how to work with VINA and set the VINA docking simulation or docking analysis. And I told us that the first thing that we need to do is to get our ligand from the database. I want to open my note once again so that's able to follow. We get our ligand from the database. And once we get our ligand from the database, we can either get it from PubChem. If we get it from PubChem, we get it in SDF format in 3D. But if the 3D is not available, I told us that we can get the canonical SMILE. Then once we get the canonical SMILE, then we can convert that canonical SMILE back to structure by using ChemSketch. And if we need to, maybe want to optimize the structure of that particular ligand, then we can use ChemSketch to add any other functional group that we need to add to it and save it back in 3D optimized format. So after that, we have been able to prepare our ligand. Also, we need our protein. The structure of our protein, we need that one too. So our protein, we have to go, either we pass through from a UniProt, we search the name for that particular protein. If the structure is available in an experimental form, we get that one even from the UniProt. And if the structure is not available, we have what we call AlphaFold. AlphaFold is a kind of simulated structure that was developed by in conjunction with Google. Google was the one behind the AlphaFold. So we can get the simulated structure for that particular protein. But if the simulated structure is not even present, maybe it's a kind of a microbial protein that is not common, and the AlphaFold has not documents anything on it, then we have to follow the step-by-step tertiary structure prediction that we were taught even last class also. So we have to do homology modeling for each of those protein in order to get their 3D structure, to get the 3D structure. So once we are able to get the 3D structure for the protein, okay, it will be in PDB format. Okay, so if it is a computationally modeled structure that we obtain either from homology modeling or from AlphaFold, then there is no need of cleaning, because it will exist even as a clean file, because there won't be any crystallographic water, and if we model it, they will be in a single monomeric state, especially the one of AlphaFold. But sometimes if we model using what is it called now, using a switch model or Fires, and we use a particular template, if that particular template that we use maybe is a kind of a dimer, okay, our model structure also will exist as a dimer. So in one way or the other, we have to clean it, because we can only use one chain to perform the docking. We can only use one chain to perform the docking, okay? And let's say in a case where the two chains that bind together, they are not of the same, size. So that means you have to first perform docking on one side and perform docking on another side. Or we try to determine which of these two subunit of protein consists of the active sites, if that is our focus, or the one that consists of a regulatory site, if that is our focus. Because it's possible that one subunit will be the one that has the active site, while the other one has the subunit regulatory site. So we have to find out that. But if they are of the same size, then we can only use one of the chain, because whatever we obtain from the first chain will be a replica for the second chain. So, and our ligand also, we have to convert the ligand. If our ligand is the SDF, is in SDF format, then we have to convert to PDB. We have to convert, because Autodoc 2 cannot take SDF file for a change, so you have to start from PDB. So we have to convert our ligand from a SDF format. We have to convert it to PDB by using PyMo. Okay, convert it by using PyMo. Also, our ligand, if we are the one that, okay, we have optimized by using ChemSketch. Okay, the basic default format for ChemSketch structure is MOL format. So we have to still convert that particular MOL to PDB by using PyMo. Okay, there may be other software can use for conversion, but I prefer using PyMo, because if you use like an open Babel, sometimes it used to give us an error. So that's why I don't like recommending open Babel software. So it's better you use PyMo. Immediately it will convert rightly for you to PDB file. So once we have our ligand in PDB file and also our protein, we have been able to clean if it is experimentally derived structure, then we have to clean the crystallographic water. Existing ligand that may be binding to the active site or regulatory site, we clean them away. But any necessary cofactor that is good for that particular protein, then we can still retain them. And once we have done that, the next thing we need to do is to convert those files, the one of the ligand and the one of the protein. We have to convert them to what we call PDB-QT formats, because that is a file format that Autodovina recognized. So we have to use Autodoc tools to convert those files. The one of ligand, we use Autodoc tools to convert it even to PDB-QT file, and also the one of our protein. Okay, we have to also convert it to PDB-QT file. Okay, but before we convert that one of protein to PDB-QT file, we have to add what we call polar hydrogen. So polar hydrogen, what polar hydrogen will do for us, okay, all the side chain amino acid that has all the possibility of having forming hydrogen bonding. So that one, we add hydrogen to them in order to, okay, by the time they want to react with the ligand, we have a kind of a means of doing that. So once we have been able to add hydrogen polar only to our protein, then we can save it in PDB-QT. So once we have saved it in PDB-QT, Then we need to generate what we call a grid box. That is, we want to find a portion of our protein where the docking will take place, And I told us that, okay, if we are familiar with that particular protein and our analysis want to focus only on the active sites, then we have to determine the active site either by reading the literature. Okay, we read literature that support that particular protein. Okay, even from the UniProt page of that particular protein, they will have mentioned 1 or 2 amino acids that are present at the active site of that particular protein, or we read a publication that support that particular protein. So once we can identify the amino acids that are found even at the active site, then we can be able to select those amino acids on the interface of Autodoc tools, then now set our docking grid box on those sites only, so that we know that this is the area that we want this analysis to take place. But in majority of the analysis that is being done, we assume that, okay, this particular ligand may not necessarily binds even to the active site. It may be the one that binds even to the regulatory site, because based on the types of inhibition, not all the inhibitors bind to the active site. Some even can bind after the ligand has already binds to the active site. So based on that kind of a background, so we try to, okay, do what we call a kind of a blind docking. That is, we assume that the ligand can bind to any part of that particular protein that will give the best binding affinity. So to do that, what we're going to do is to extend our grid box to cover that particular protein, in every means, and that one we can do it even on the interface of Autodoc 2 by extending the XYZ dimension for us to cover the protein structure appropriately. And I told us that if after we have extended the XYZ dimension box and we still not able to cover the protein structure, then we can increase the spacing. The spacing will enlarge the box so that we have more of the protein to cover. And if we are having excess space also, we have to reduce back the XYZ just to ensure that we don't have excess space and we don't leave any part of that protein uncovered. So immediately we have done that, the dimension of that particular grid box will be saved out as grid.txt in a TXT file, in a folder in our working folder. We save it there. So that particular grid dimension, TXT, contain all the information about the grid box, and that information is what Vina need in order for it to work on the binding, so that we know that, okay, this is the dimension where the ligand has to be sampled and binds to in order to calculate the necessary binding affinity. Also, I told us that once we have been able to prepare our ligand in PDBQT, prepare our protein in PDBQT, and we generate the grid.txt file, then the next thing is we have to use Autodoc Vina software, which is the software that will do even the redocking. So Autodoc Vina software, since it doesn't have what we call graphical user interface, okay, doesn't have graphical user interface, then we have to run it from the command prompt. I told us that any software that doesn't have graphical user interface, then we have to run those softwares from the command prompt. And I told us also that all the files that we want to run must be in the same folder with that particular application. Our case here is Vina software. Okay, so all the file that you want to run by using this particular Vina must be in the same folder where Vina is located. So it's not that the file ligand PDBQT, protein PDBQT, and your configuration is another folder, and your Vina is another folder. It will not recognize it. So your ligand, your protein, your configuration file must be in the same folder where your application is found. And I told us that we have to copy it from the program parts, because sometimes program parts require administration permission, which by the time you are running your docking, you will not be able to recognize that. So in order to avoid administration permission during your analysis, we have to take that particular Vina folder out from the administrative part, So we put it on the local disk, directly. So, and I told all this how the grid text look like. It will have information for that particular protein or your enzyme that you have generated that particular grid dimension for. It will have the information about the spacing for that grid box. It will have the size of that particular grid box, and then it will have what you call the center of that particular grid box. So this grid.txt is peculiar per protein. If you are working with 2 or 3 proteins, you have to generate the grid box for each of those proteins. You can't use the grid dimension for one protein for another. It will not work. It will not. So by the time you are doing your preparation with Autodoc 2, then the protein, when you get to that particular protein, you ensure that you generate the actual grid parameter for that particular protein. So it's that one that we now use by the time you are running your Autodoc Vina software. So this is how. So that grid, this particular grid.txt will now serve as a basis for us to populate our configuration test for Vina. Vina make use of a configuration test. Okay, so the information that we have in our grid.txt file will now be what we use to populate Vina configuration file. And this is how Vina configuration file looks like. It will have the receptor name. Okay, that is a protein. It's okay. So the name of that particular protein that you have inside the Vina folder. Okay, for this particular one, the name of the protein is afpdbqt, but that is a file format. If you put a .pdb here, Vina will not recognize it. So it has to be .pdbqt. Then our ligand, which is the second thing inside the configuration text, the name of that ligand that you want to bind with that particular protein at that particular point in time. So we must have the name of that particular ligand in your configuration file. So it must end also with a pdbqt. Then about the box, about the grid box, the center, you have to copy directly from the grid.txt file that you have generated. Just copy it and put it in front. For center, which is the one of we have it center over here. So I just use another number in the one that you are seeing on your screen. So you put it, the first one is X, the second one is Y, the third one is Z. Okay. Then for the size, that is a NPTS. Sometimes you could make a mistake, maybe you are in a rush. You can change the center for size. By the time you want to run, if you say it will not run, it's giving you error. So you have to ensure that, okay, you use size for where size is supposed to be, and you use center for where center is supposed to be in the Vina configuration file. Then we have what we call output. Output, that is the output file that Vina will generate for you. So you have to name it. Because if you don't name it, it will continue to overwrite. By the time you continue to run, it will continue to overwrite each other. So you have to name it per run. So if you run a particular ligand 1 against protein 1, name it ligand 1 against protein 1 .pdbqt. So by the time you want to run for another ligand 2 against protein 1 .pdbqt, so that the output will not override each other. So the basic formula so that your work will be spelled out is, okay, use the name of that ligand you want to run underscore the name of protein that you are running it with, then end it with a .pdbqt. The exhaustiveness is the extent by which the Vina app will try to conduct even the binding on each of the cavity. Then we have the spacing. That one is also from the grid.txt file that you have generated. So the spacing, you put the value there. So having done this particular configuration file for Vina, the next thing is for us even to run Vina. And to run Vina, we have to implement the running from the command prompt, as I have said. So whatever be the path that you are when you open your command prompt, you have to navigate back to the local disk. That is where we have positioned even our Vina folder. That's where we have positioned our Vina folder. So we have to change the path even to that place where we have our Vina folder. So to move back or to move back from the folder where you are in your command prompt, when you open your command prompt, you have to type cd dot dot, no space, cd dot dot. Then you press enter. This one will move you a step back. So if you are yet to get to local disk, which is a C directory, then you have to press cd dot dot again until you now get to the local disk, which is your C directory. Then after that, to move into the VNAV folder, to move forward now into the VNAV folder that you have inside in your local disk, you press cd space VNAV, and then you press enter. Cd space VNAV, then you press enter. So this one will move you into the VNAV folder where you have all your VNAV app, VNAV split app, then all your ligand, PDBQT, protein PDBQT, configuration, .txt, PDBQT, where you have all those ones. So to implement VNAV to run the binding for the configuration information, that you have, then you have to run this particular command on the command prompt interface. VNAV space -- config. Then the name of that configuration file is a conf.txt. Then you press enter. So if everything is okay, then it will start running and you will see all the processes showing on your screen. So depending on the size of the ligand or the size of the protein, that one will determine the extent, the time that it will take for it to run. If your protein is so big or your ligand is so big, then it can take minutes, more minutes. But if they are the size of those protein or ligand are small, then within a minute or 30 seconds or 2 minutes, you will get the score. The score will be displayed on the screen, and it will rank it based on the binding affinity. The more negativity, the better. Okay, so it will rank it based on the binding affinity. Okay, so the first one that has the highest negativity will be the one on top, and we often go with that particular one. But at a point in time, let's say for example, you want to see other portions apart from the first for the first one that gave you highest binding affinity, then you can, even from the result that you obtain here, if you still want to check, okay, what I want to focus upon is not regulatory site, it's still the active site. So you can visualize all the binding affinity posts that he gave to you. You can visualize it. So by the time you visualize, you'll be able to, okay, this is the one that I want to take because based on my study, this is the area that I want to focus upon. So immediately we generate this particular result. Once it's finished and you have the result on your screen, you have the, what is it called, and the binding affinity on your screen, then you pick the first one, you write it down. It's possible to copy all the results from the screen and just go and paste it in your Microsoft Word for later reporting. So we have to split the output file to all the numbers of a result that we have on our screen. If you have 9 there, 9 binding affinity, so that means the ligand can bind to different 9 poses on that particular protein. So we want to visualize each of these posts in a different way. Okay, we want to separate them instead of being clustered together like that. So to do that, we have to use the second app in our folder, which is a VNAV_split. Okay, so the command to initiate that particular operation is VNAV_split, which is the name of that particular app, space -- input, because now we want to input something from the file. And what we want to input is the output file that we generate from the first implementation. Okay, and the name of this particular output file will be the one that you have put inside the configuration file, here. Okay, that is what it will have been written out for you inside of VNAV folder at the end of the first implementation. So you just copy the name of that particular output file. Then it must end with PDBQT because the file is already in PDBQT. So you just copy the whole name and then you put it there and you press enter. So once you press enter, you will not see so many things, just like about two line report on your screen, but the actual splitting will be inside your folder. So by the time you go back to your folder, it will have splitted this output file into the number of binding poses that your ligand that the ligand your ligand has, then you can see from there, okay, ligand 1, it will be a different file, ligand 2 different file, ligand 3 different file, to the number of poles that you have from your first implementation. So for us to visualize this file is very easy. We can use this Autodoc the Autodoc tools that we use to prepare our PDBQT. You can use it, but somehow it will not give you a clearer picture. Okay, so for us to generate the 3D file for our visualization, we use Pymo again. So you just will open your Pymo software again, then go to File, then you open. So open the protein. You can either use your PDBQT that's inside your VINA folder here, or use the one of PDB that you have clean. But I prefer you using the PDBQT since you are working in the same folder, VINA folder. So use the protein PDBQT, open it with your Pymo. So once that one is open, the protein will be there on the interface of Pymo. Go to File again, click Open. Then open the ligand from your VINA folder, the ligand 1. If you want to visualize the first ligand post, you have to click on ligand 1. It will be, the name will be there, 1 will be there, 2 will be there, 3 will be there. So click on ligand 1. So once let's say now want to visualize the one for ligand 1, just click in your Pymo, File, Open, then you navigate to your VINA folder, then you open the result that has ligand 1. The one that has ligand 1, it will also be in PDBQT, ligand 1. So you open it. So Pymo will open the ligand for you, and the position where that particular ligand binds to on the protein, it will be there. So it will no longer be separate alone. It will now bind to a particular cavity on that particular protein, which you can easily see. Okay. So on the Pymo, there are some buttons beside it where you were able to, okay, if you want to see, okay, which amino acid residue even surround this particular ligand. Okay, this particular ligand is interacting with alanine, with glycine, or the rest. So there's a button where you can do that, which is very easy by the time you play around it. On the right hand side, you see some buttons there, C, L, R, and the rest like that. So also we can see, okay, you want to visualize just only the cavity where that particular ligand is. Then by the time you click on all, you see all on the top, on the right hand side of Pymo. There, just click on the first, I think that one is C. So once you click on it, go to what you call presets. By the time you go to presets, then it will give you another option. Then go to ligand site. Ligand site. By the time you click that, give you another option. Okay, how do you want to represent the ligand site? Is it as a cartoon or as a string and the rest? So click as a cartoon. So it will zoom where the ligand binds to and show you that cavity. Then since you have, okay, I want to see the amino acid residue that surrounds the ligand, that one also will show you the amino acid that surrounds that particular ligand. So that's a kind of a 3D view using Pymo. So you can report that particular interface, just change the background, go to display. At the top of Pymo, go to display to give you options. Just scroll down, you see background. Then you can change the background from the black, which is the default, can change it to white. So that if you want to report that particular interaction, the background will be white background. Then you can snapshot it and then save it for your report. Also, this particular web server, www.dxu.org.software/software, has a kind of application that you can use to visualize even our docking results, and it will give you a good, bright future. It will show you the pi stacking, hydrogen bonding, and the rest like that. So it's very good. That's why I put it there. So by the time you visit this particular site, www.dsulab.org/software, it will give you the first interface or the different EZ leak plot. So once you click on EZ leak plot, you can only use this particular software on desktop interface. Even if you are working with phone, you have to configure your phone back to desktop interface to visualize it. If you are working with computer, it will always be in desktop interface. So once you click on that easily plot, it will open another interface for you where you have to agree. So just click agree, and after that, it will open a blank page for you. On top of that blank page, you will go to the top towards the left, you see application. So once you click on application, it will still show you all the list of the software over there again. Then click on the leak plots. Click on EZ leak plots. So once you click on that, it will give interface for you where you can upload whatever you want to visualize. The first one is okay, upload your protein. Then the second one will say upload your ligand. So for the upload of protein, go to your protein that you have used to conduct the docking in your VINA. Okay, go and open that particular protein in your PDBQT file. Okay, in PDBQT format. Okay, open that particular protein, navigate to VINA folder, then click on that particular protein.pdbqt. Then for the ligand, so the ligand one is the one that has the highest binding affinity. We want to visualize that one. Anyone that you want to visualize, you can upload that particular one per time. So let's say now I want to visualize the one for ligand 1. Okay, then just go to under the ligand upload, navigate to VINA folder where you have your results ligand 1.pdbqt to upload that particular one. Then if you show you, okay, how do you want to visualize this particular interaction? Is it in 2D view or 3D view? So I prefer using 2D view because 3D view will not give you a clearer information, a clearer picture. So I prefer using 2D view since I use PyMode to generate my 3D view. So you click on 2D view. So you now tell you, okay, how do you want to save it? PNG or SCSV or so. So just click on PNG because you want to save it as a picture. Okay, so once you click on PNG, then click start. So within seconds, maybe 30 seconds or less, it will generate the interaction between your ligand and your protein. It will generate it for you and show it to you on the screen. So once you see it on the screen, to download it on the left-hand side towards the bottom of that particular picture on your interface, you will see save. You will see save. So just click on that save. So once you click on that save, it will download that picture into your download folder on your computer. Okay. So, but it will not name it. So you have to go, if you want to do as many as possible continuously, you have to go immediately to that your download folder, go and name it with the name of the protein and the name of the ligand that represent that particular picture. Because if not, it will just show you output. So it will not give you any. So if you just lose focus and you not go and rename it immediately, you may not know which kind of interaction is this particular one, especially if you are working on a large amount of data. So immediately that one download into your download folder, just go to the folder, then go and change the name to the right interaction, ligand 1 against protein 1. Just write the name there. So because if you run another one, it will show output 2. Output 2, output 3, like that. So, but you have to give it a name so that you're able to know the actual one that it is. So that is about the overview of Autodoc VINA, and it is very straightforward if we can just follow all these steps. Thank you.